Good morning, everyone. I am Vaibhav Rajan. I am an assistant professor in the Department of Information Systems and Analytics. Thank you for this invitation. Today, I will be talking about an aspect of healthcare analytics that is of interest to many of us in my group, which is learning from multiple heterogeneous data sources. And these data sources include the more traditional data sources like clinical data from hospitals and data from clinical trials. But increasingly, they also include data from smartphones, wearable devices, and search engines, which are telling us about various healthcare trends as well as giving us more information about individual health. And all this data presents tremendous potential for both enhancing classical applications like diagnostics or population health, and also presenting new opportunities for things like precision medicine and for tremendous optimization and efficiency improvement of hospitals and so on. Let me give you two specific examples. The first example is that of disease gene prioritization. A disease gene is a gene with disease-causing aberrations. Biologists and clinicians want to understand the mechanism of diseases in order to improve diagnostics and develop better treatments. However, proving a causal link between a gene and disease experimentally can be expensive and time-consuming. As a result, what is often done is to prioritize candidate genes before experimentally testing them in the lab. And this can drastically reduce the human labor involved and the costs. So how one usually goes about it is, for a given disease, right, one starts with a list of candidate genes, and data about this gene with respect to the disease, data about various genes here are collected and features are extracted from these data sources. We need not go into the details of all these features, but what is important for the purpose of this talk is to realize that these features essentially give us evidence of various kinds from different data sources: functional evidence, cross-species evidence, mutation-based evidence, text evidence, and so on. And using all these different data sources, one may want to come up with a kind of scoring function that can be used to rank the input gene list. So that ranking provides the prioritization that can be used to determine which genes to test. Take another example: ICU mortality prediction. Intensive care units are where the most critically ill patients are housed in a hospital who require continuous monitoring and care. ICUs are very expensive. They represent about 22% of the total hospital costs, 5% of the total healthcare costs in the US. And the need for ICUs has grown worldwide, especially in today's situation. But resource availability is limited. Resources both in terms of personnel as well as equipment. So there is a need to prioritize care within the ICUs. And this is done by assessing the severity of patients and by identifying high-risk patients through various risk calculators. Mortality risk is one of them. Once you have such a risk calculator, then that can be used to aid critical decision-making and to enable patient triage or prioritization, thereby making resources available to patients who are more in need of them. The data in the hospital, it's best viewed when you think of a patient trajectory. So I have a schematic here that shows how the patient goes from one unit to another And while the patient is there, different kinds of data are collected for the patient. They can be vital measurements like blood pressure, heart rate, etc., various lab investigations, medication orders, nursing notes, various scans like MRI, CT scans, and so on. So all this data uh, can be utilized to identify whether the patient is at risk. All this data is typically in the electronic medical records within the hospital. And again, the task is to get some kind of patient representation or a feature vector which can be used within a predictive model. So in both these examples, we saw how one needs to learn from heterogeneous data sources. So representation learning is the field of learning effective representations from various data sources. In this case, we wanted to learn gene representations. In the other example, we wanted to learn patient representations that can be used in predictive models. Now, this is a key challenge in big data analytics, not just in healthcare, but in other domains as well, because manual feature engineering becomes impossible as you have more and more data sources, and also with increasing heterogeneity. The heterogeneity can be in terms of modalities, like you have text and images and videos and structured data and so on. Right? Even within structured data, there can be heterogeneity in terms of the data types. There can be heterogeneity in terms of the temporality, as well as sparsity, noise levels, and so on. So for the purpose of this talk, we will assume that we're dealing with relational data. So we'll think of data being contained in matrices that contain pairwise relations between entities. So these entities could be things like patients, medications, labs, and so on. So if you take, for example, the patient medication matrix, the ijth cell contains some data about the jth medication given to the ith patient. And this could be different in different contexts. It could be binary, whether the data was, whether the patient was given the medication or not. It could be the dosage of the medication as a real valued number, and so on. So representation learning has been studied in other contexts, and we'll do a very quick overview of two other contexts where they have been used in recommendation systems and in multimodal or multi-view learning. So let's quickly switch context and think about the recommendation problem. Here again, you have a matrix, which is the user rating matrix. The rows correspond to users, the columns correspond to movies, and the cells have the ratings that are given by the users to the movies they have watched. Many movies have not been watched, and hence this is a sparse matrix. And so we assume that there is a low rank factorization, which can give us, which can decompose this matrix and give us a user factor matrix and a movie factor matrix, U and M. And this gives us a user-specific representation, which is of lower dimension, as well as a movie-specific representation, which can be combined to reconstruct the sparse user rating matrix. So that is the key idea that we should bear in mind, and we'll see how this is used in different contexts. Right? So the optimization problem becomes that of minimizing the difference between the reconstructed matrix and the original ratings. So we find factors U and M such that this reconstruction loss is minimized. When we have additional data about the users or about the movies, that can be used as side information and can be brought into the optimization framework very easily, pre-multiplying and post-multiplying by these matrices. So the idea has been used before in the case of gene disease association. So you now, switching context back, we think of instead of users and movies, we think of genes and diseases here, right? And the user rating matrix can be thought of as the gene disease association matrix, right? So whenever there is an entry in the matrix, it basically tells us about the association. And the problem is very similar, because here also you have a large matrix. It's very sparse because most of the associations are unknown and so on. But here we have a wealth of side information that can be used. So we have information about the genes and information about the diseases that can be used within this optimization framework. And this is what was the key idea behind inductive matrix completion, which was used to predict gene disease associations. Most state-of-the-art recommendation systems use neural architectures, and many of them use autoencoders to obtain these representations. And as many of you would know, autoencoders are simple, the vanilla variant is a simple feed-forward neural network that takes in an input X and reconstructs X prime in such a way that the difference between X and X prime is minimized. And it passes through multiple layers, where the bottleneck layer has a lower dimensional representation of the input. So the first part of this network is an encoder, and the second half is the decoder. So similar autoencoders have been used in various ways in recommendation systems. So here is one example where the user factors are essentially coming from an autoencoder. Similarly, movie factors are being learned through an autoencoder. And then the factors are multiplied to obtain the user item rating matrix. Now in multi-view learning, so we going from one matrix in recommendation systems to 3 matrices when you have side information, and now we come to multi-view learning, where we have multiple views of the same set of subjects. So think of the subjects here as patients, and you have multiple views in terms of the medications given, lab investigations, diagnosis, and so on. If the views have different modalities, then this is called multimodal learning. The fundamental problem remains the same, that of getting good patient representations from all this different data. In multimodal learning, again, autoencoder based architectures are commonly used. Here is an example which shows an audio input and a video input, which is together used to obtain a shared representation of an entity. So augmented multiview learning essentially generalizes this to an arbitrary collection of matrices. Right? So here the problem is to learn representations of each of these entities, each of the entities that are involved in the input collection of matrices, in a way that knowledge can be used, knowledge of all these matrices are used to obtain that representation. So here on the right side, we see an example with 5 matrices and 6 entities. So this is just an example, but it can be any collection of matrices, including the collections we saw in the case of recommender systems and multi-view learning systems. So it's basically a generalization of that setup. Collective matrix factorization essentially aims to do just what I said, learn representations of these entities jointly from the M input matrices, right, such that each entity has a unique representation. And this is done in a very similar way. I mean, it's just a generalization of the matrix factorization setup we saw, where we want to minimize the reconstruction loss of each of these M matrices with respect to the learned factors. So the factors are learned in such a way that they learn from all the matrices that contain a particular factor. So for example, E1 here, the entity E1 is present in X1 and X2. So the factor UE1 is basically learned from all the matrices together and used to reconstruct the same UE1 is used to reconstruct both X1 and X2. And because this is done jointly, information from all the matrices are used to learn the factors. So if we look at previous models, deep learning based models have improved both multi-view, classical multi-view models as well as recommendation models. because of their ability to model nonlinear structures within the system of matrices that's being modeled. But deep learning based models that have been designed, that were designed for multi-view models, they do not generalize easily to more than 2 views. And recommendation based models, deep learning based recommendation models, they have mostly been designed for these L-shaped configurations where you have the user rating matrix and side matrices. CMF is the only model that can generalize to all the to any configuration of input matrices, but there's no deep learning based architecture there. So our goal in this work was to explore deep learning based architecture for collective matrix factorization, and we wanted to see if that can be used to if deep learning can help in improving representations learned through collective matrix factorization. So the aim, as usual, as we saw, is to obtain low rank factorizations of M input matrices jointly, where each entity-specific factor is learned from the input data through a nonlinear transformation, which is parameterized by the weights of the neural network. So this nonlinearity is essentially modeled through a neural network. So we assume that we're given a collection of M matrices and a graph which denotes the relationship between the entities and matrices. So just to give you an example, we have the same example that I had shown you earlier, 5 matrices, and the 6 entities. The graph essentially shows what are the entities that are associated with each matrix. For example, X5, as you can see, X5 is a relationship between entity 6 and entity 3, and that's what the graph shows. Okay. So how is the DCMF model constructed? There are 2 steps. First, for each entity, we need to concatenate all the matrices that have information about that entity. Well, concatenation is not the only way, but this is we started with the easiest approach. There are other ways to do it as well. And then we obtain entity-specific latent factors through the use of autoencoders by using this concatenated matrix as input to the autoencoder. Now, all these autoencoders are trained together, and the optimization problem is to minimize both the matrix reconstruction loss as well as all the autoencoder losses. So remember, this is dynamically constructed because the input can contain any collection of matrices, which means the input can have any number of entities, and depending on the number of entities in the input, we dynamically construct a neural architecture that can that has the appropriate number of autoencoders. So just to give you an example, for the same collection of matrices, 5 matrices here, consider entity E1. So E1 is present in matrices X1, X2, and X3. Right? So here we show the 3 matrices in shaded here in these 3 here in the smaller picture here. And the concatenation step essentially concatenates data in these 3 matrices and passes it through the autoencoder. And U1 is the entity representation for entity E1. Similarly, for entity E2, which is present in X4 and X1, the corresponding matrices are concatenated and passed through the second autoencoder and so on. So all the 6 autoencoders and the 5 reconstruction losses corresponding to the matrix completion are used together, and the entire model is trained together. So the model is relatively straightforward. So the main difference between CMF and DCMF is that in CMF, you learn these factors directly such that when they are multiplied, they recover the mth matrix. Whereas here, the factors are learned through a neural network, the encoder, on the encoder which essentially learns this using the concatenation of all the matrices containing that particular entity. The challenge really comes in the optimization, because the matrix reconstruction losses are dependent on multiple autoencoders, and generalizing to arbitrary collections of matrices is very difficult. The problem arises due to varying dimensions in the input matrices, or and varying sparsity levels of input matrices. So just to see why this happens, consider an example where E1, we want to learn the representation of E1. So we start by concatenating the matrices that contain information about E1. So let's call this C, and let there be P samples here, and let the dimensionality be Q. So if P is greater than Q, then essentially the dimensionality of C is low and the number of samples is high. So there is a possibility of overfitting here. On the other hand, if Q is greater than P, the dimensionality of C is high and the number of samples is low. And there is a possibility of underfitting. Now, in both cases, we know how to deal with it if we have a single network. If it's overfitting, we can try things like regularization, early stopping, and so on. If it's underfitting, we can try things like pre-training, increasing the number of hidden layers, and so on. However, the challenge here comes because the hyperparameters can differ for each autoencoder, and we don't know what these hyperparameters should be because they are dependent on the data. The C matrix is being constructed by using the data, and the data can be different each time. So essentially, manual tuning is infeasible, and we have to determine both the structure of the network as well as the hyperparameters dynamically. Similar problem arises due to varying sparsity levels. If you have a dense autoencoder encoding, then it favors the reconstruction of dense matrices and not sparse matrices. If you have a sparse autoencoder encoding, then it favors the reconstruction of sparse matrices and not dense matrices. So essentially, this led us to think about hyperparameter optimization here as a way to deal with these problems. And there are many hyperparameters. They're model related, like the entity dimension, number of hidden layers, and so on. There are also optimization related hyperparameters. And of course, the standard way to deal with this for any large neural network is to use automated hyperparameter optimization. So we want to find the optimal hyperparameters for a particular model, right? But note that the model is the loss function, if you think of it, is in terms of the weights of the network. It's not in terms of the hyperparameters. So the functional form of loss with respect to the hyperparameters is not known. But it can be computed. For any hyperparameter set, you can train the model and compute the loss. Right? So this is essentially a black box function optimization problem, where you have you can run you can get the loss values for a number of different hyperparameter sets, but the problem is you want to find the best possible hyperparameters. And how do we solve this? Those of you who are familiar with Bayesian optimization will know. Essentially, we also use a model, use machine learning. Right? So we fit a surrogate model to the data, but we do it but we use sequential optimization here. So we want to sequentially explore the space of hyperparameters in such a way that we explore a little bit and then exploit the knowledge that we know about this space to lead our way into a direction that takes us to a good hyperparameter selection. So this is done through what is called an acquisition function, right? And they're designed to have optima at points with high uncertainty in the surrogate model, such that they facilitate exploration, or they have optima at points with high predictive values, which facilitates exploitation of what is already known. I'll skip the details, but for this model, we developed a variant of the expected improvement heuristic, and we use a multitask Gaussian process to as the surrogate model that more details can be found in our paper. So to summarize, given a set of input matrices and the entity matrix relationship graph, for each entity, we basically concatenate the matrices that contain information about the entity, and we construct an autoencoder, which is used to learn the entity representation. Then we we train this network collectively by minimizing both the autoencoder loss as well as the reconstruction loss. Bayesian optimization is used for hyperparameter tuning. And finally, we obtain a representation of each entity. We can use these representations to complete the matrix. So we tested this approach for the task of gene disease prioritization. We have 5 matrices here, and they contain 6 different entities: genes, diseases, patients, patient features, disease features, and so on. So all this data is collected from publicly available databases, and we restricted the study to breast cancer genes here. So the task is to predict gene disease associations that are known, and we use the standard 5-fold cross-validation of known gene disease associations to evaluate the models. So the main baseline is inductive matrix completion that I mentioned earlier. This is at that time the best known method to predict gene disease associations. We also used CMF and a variant of CMF called group sparse collective matrix factorization. For each disease in the test set, the genes are ranked by the score predicted by each method. And we use a standard evaluation metric called probability at n, which is the CDF of the rank, so the probability that the rank at which a hidden gene disease pair is retrieved is less than a threshold n. It is similar to recall it. So our results showed that DCMF significantly outperforms all the other baselines, which goes to show that the model can effectively use the nonlinearity that can model using neural networks. At values less than 10, also it has competitive performance. So nonlinearity that is modeled through DCMF is actually helping in obtaining better representations. So more details and other experimental results, including on recommendation problems, are discussed in our paper. And this is work done with Raghunathan Mariyappan, a research associate in my team. So we are using the same kind of framework for another problem, related problem of combined data-driven knowledge-based modeling. So we know now that CMF-based models like DCMF can obtain patient representations from an arbitrary collections of data matrices. So these matrices need not be just data that is collected from a particular hospital or clinical data of some sort, but they can also be any sort of auxiliary information that can help in getting better representations. So the primary source of knowledge in biomedicine is from randomized controlled trials and case studies that are all recorded in the literature, biomedical literature, in journals. And there is a lot of ongoing research to automatically create knowledge graphs by mining the biomedical literature through various kinds of NLP techniques. So these graphs essentially contain a large, very, very large number of nodes and various kinds of edges. So it's essentially a heterogeneous information network where there are different kinds of nodes. So for example, a drug and a disease Right? They can be two nodes and they can be connected by an edge which indicates side effect. Right? So the disease is a side effect of the drug. Or they can also be connected by an edge like treats. So the drug treats that particular disease. So all this kind of information is encoded there. And of course, one way to use it would be to just take the adjacency matrix from this large graph and throw it into CMF. But that, although that helps, it does not help. It causes problems due to the scale and sparsity of the adjacency matrix. Another way to go about it is to use graph embeddings, which are vectorial representations of the nodes in the graph, in this case, diseases or drugs and so on, that can encode global structural patterns that are found in the entire graph. So these graph embeddings can be used as inputs and can be added into CMF-based models very easily to obtain better representations. So we've also been working on obtaining representations of such kinds, and our initial results show that in several tasks, these additional sources of knowledge actually are very helpful, and they improve the representation significantly. And this is also the case when you don't have enough samples. So for example, rare diseases, right? In case of rare diseases, even if you have a large clinical database, there may be very few patients who have a particular rare disease. Whereas information about such rare diseases can be found in the biomedical literature and can be used within the modeling process very easily through CMF-based approach. Right. So to conclude, we developed a deep learning, and we believe this is the first deep learning based architecture for collective matrix factorization that can seamlessly integrate heterogeneous data sources to learn effective representations using neural networks. And we believe this can be a very easy to use framework for developing combined knowledge-based data-driven models. So the representations that are learned from DCMF can be used for various kinds of predictive analytics. For example, in gene disease prioritization, that can be used in turn for diagnostics and treatment development. They can be used to develop various kinds of predictive models like ICU mortality prediction, which can help in patient triage and better resource utilization in hospitals. So heterogeneity, that was the topic of my talk, is found not only in different kinds of data, right, in different modalities, but also in the way we find temporality in clinical data. So data within the hospital is often temporal because there are repeated measurements of the same data type. For example, blood pressure is measured multiple times for the same patient. But these different measurements vary considerably in their scale. So some measurements are taken more often, some measurements are taken, they can be very sparse, and some can be very high frequency data as well. Right? So combining all of these together is also a challenge. Right? Additionally, they can vary from one patient to another. There are other challenges, for example, challenges due to noise and uncertainty. There may be recording biases. Sicker patients are often measured more often. Right? And on the other side, from an application use perspective, there's very high accuracy requirements for predictive models in healthcare. Because there are high stakes involved, and there's also a need for interpretability. And lastly, as I mentioned earlier, there is a very large source of biomedical knowledge, which if we know how to utilize within our models can be well infused with such knowledge, and can hopefully be more interpretable as well as more accurate. Right. With that, thank you very much, and I'll be happy to answer any questions.